Welcome to Wildcat Science with Mrs. Haugo. This is biology and we are studying genetics and we are looking at activity number 19 today. So as we talked on Friday, activity number 19 is biofarming edible vaccines, and the challenge is what are the benefits and trade-offs of genetically modifying crops to contain edible vaccines. So what we already previously talked about was edible vaccines, making the edible vaccines, and vaccines are important for fighting diseases and they make the person immune to the disease. And also, what you have to do is you have to isolate the DNA, and then you isolate the correct gene and make lots of copies of that using polymerase chain reaction. And there's a nice picture of PCR on page 396 where the section of the gene that you are looking for is copied in the first round, and then you have 2 of them, and then 4, and then 6, 8, and 16, and so forth. And then we make a DNA construct, and then we put the DNA construct into the organism we want. There's a nice picture of making a DNA construct on page 397. So it has a picture of a starter and then our virus protein gene that we are interested in for our vaccine, and then an antibiotic resistant gene. And the reason for that antibiotic resistant gene is only so when we put that virus in a bacteria, then we grow the bacteria on a agar plate with antibiotics on that, and so only the bacteria that grow we know have the antibiotic resistant gene and our virus gene, so we know which ones have the gene that we put in there. Also, there's some nice pictures on page 397 of how we can put our DNA construct into the organism we want. There are a couple of options. We basically fire them into the plant cells with a gene gun. We could put the DNA plasmid into a bacteria and then infect the plant cells with the bacteria. Or we could put our DNA construct into a virus and then infect the plant cells with the virus. The next step is the genetically modified organism, like a bunch of spinach, grows up. Then we test the organism to make sure it has the desired gene. For instance, on page 398, there is a picture of a genetically modified corn plant that had a DNA construct put into it. And then there is a gel electrophoresis that was run. And you can see on page 398 that there are some bold lines during the gel electrophoresis that shows us that our DNA construct did make it into the plant. Then the next step is to test to see if the vaccine works. So what happens is mice were fed the lettuce, the genetically modified lettuce, and then they were infecting them with the disease to see if they got the disease. So if this were the coronavirus and they had a vaccine for the coronavirus, they would first have to modify mice to get the coronavirus because mice currently are immune to the coronavirus. And then they would have to get the vaccine and grow it up in lettuce and then feed the lettuce to the new mice and then see if they got sick or not. And infect them with the coronavirus and then see if they got sick. So there are several benefits and trade-offs to edible vaccines. So the benefits are you do not have to refrigerate the vaccine. So we know that getting refrigerated vaccines in certain parts of the world can be a problem. But the lettuce, you would grow it and then eat it. So that could be a real benefit in some parts of the world. But there are also drawbacks. So one major drawback is that the amount of vaccine varies in the plant, so you might not be getting enough. You could say, well, everyone's supposed to eat a cup of lettuce, but maybe you only got half the amount that's in a normal cup of lettuce, so then you only have half of the vaccine. So then you think you're immune, but you're not. So that is a real problem that needs to get figured out. All right, so that was an overview of how edible vaccines are made. Tomorrow, in our next video, we're going to talk about activity number 20, are genetically modified organisms the solution? And we'll look at some data and discuss that. All right, see you then. Bye.